Welcome to Geriatric Grand Rounds, and thank you all for joining us. So, Helen John, there's a lot of anxiety introducing somebody as distinguished as Dr. John Newman. John has been a friend and a colleague of ours for many years now. I still remember when John approached us as a distinguished fellow, already a well-known scientist. Here to start where he joined us to start his fellowship in geriatrics after finishing his residency here. And this was after he had accomplished his MD and PhD at the University of Washington. And since finishing his fellowship, he's been on faculty ever since, and has a lab at the Buck Institute, so has a joint appointment between UCSF and the Buck Institute. Yet is often seen, you'll often see him clinically here. He attends on the ACE unit. You're still doing internal medicine attending at the VA?
Yeah, a little bit.
Over and well, yeah. So, John, I think you're going to spend a lot of time explaining to us what translational geroscience is, but that is kind of what John does. He's kind of become one of the international leaders of translational geroscience, and I kind of view it as like connecting the dots between the basic biologic mechanisms of aging you study in your lab and the delirium and functional deficits and dementia that you see when you're attending on the ACE unit, and trying to bring that together and helping understand through biologic mechanisms how we can prevent those syndromes and how we can help people with those syndromes live better. John has really become one of the national thought leaders in this area. He, in addition to his basic science work in his lab, has quite a reputation as an educator. He's developed a lot of national initiatives to enhance geroscience education and enhance geroscience training, and also to bring together the fields of clinical geriatrics and the biology of aging. So, John, look forward to hearing all the great things you have to tell us.
Thank you, Ken. That was a great introduction, and thank you for all of that. Having an introduction like that from the great Ken Kavinsky is high on my list of career to-dos. So I'm checking that off. Thank you, Ken. And most of you know me pretty well. Some of you may not. Everything that Ken said, I've been faculty now since 2014, believe it or not. Mostly do research at the Buck Institute, which is up in Novato, but do my clinical work at the VA and love being a part of this geriatrics community. I arrived as a wet behind the ears intern in 2008 as a basic scientist, a pure bench scientist. And I have good news and bad news for you guys. The good news is there are no Western blots in this talk. The bad news is there are no cute mouse pictures either, because this is entirely a clinical translational talk. And we're actually going to talk a lot about clinical research and clinical trials. How, as Ken said, I am trying the best I can to bring aging biology to improve the health and lives of older adults. And in a way, this is kind of representing how my career has evolved in the last 10 years. There's still a lot of mice in my lab. Not these. These are my dogs. There are still a lot of mice in my lab, but I now run a full bench-to-bedside translational research enterprise, which I'll introduce you guys to as we go through. I do have some disclosures. Over the last 10 years, I've helped to co-found or advise several companies, one of which, Component Health, I'm a co-founder and small shareholder, but they supply a compound that we're using in 2 of our clinical trials. They don't fund them, they don't have an operational role, but we're using their compound, and I'll point out where that's relevant. So as Ken said, we'll start off with what is geroscience? What am I talking about? And then I'll kind of give you how my career has evolved to become more clinical research oriented, starting from mice and going into clinical trials with ketone biology. I'm not actually going to talk a lot about ketone biology, just enough to kind of illustrate how this path worked. And then give you guys the snapshot of the present, which is really the future of translational geroscience, of aging biology being tested in clinical trials to improve the health of older adults in a really big way around the world and here in the United States. Where does this stand? And maybe, if you're interested, how could you help? So we'll start at the very beginning with aging and the idea of aging as a therapeutic target. My favorite way of illustrating the effect of aging on health, and I guess I'll say right up front, aging is not all bad. I've enjoyed a lot of things about aging already in my life. I love interacting with older adults. A lot of things get better as we get older. There are some things driven by aging that maybe we wish we could leave behind. So we're going to focus on those pieces of aging that are detrimental to our health. Aging influences health in powerful ways. My favorite example to illustrate this is the broken bone. A couple of years ago, one of the 23-year-old research associates in my lab fell off his skateboard and broke a long bone in his arm. It really made no impact on his life. He had a hard time using his pipetter in the cast, and it was kind of an inconvenience for a few weeks, but otherwise he was fine. Made no difference, really. We all know if you're an older adult, especially a frail older adult, whether or not you're skateboarding, if you fall and you break a long bone, like your femur, that is a very, very different thing. That's a life-changing event, and for many, the beginning of the end of their life. Same pathophysiology, it's just a broken bone. It's not even about the broken bone, but a hugely different impact on people's health because of the state of aging of the person. Aging is not just about how your body responds to a stress or a problem like that, but we all know aging is the primary risk factor, in most cases, the dominant risk factor by far, for most of the chronic diseases that most of us, most doctors in general, spend most of our time treating, and that most affect the lives of most of our patients, all driven by aging. We all know in this room that age is not a number, though. Your risk for Alzheimer's disease isn't exactly driven by the number on your driver's license. And we all know what many doctors haven't learned yet, that very, very few clinical decisions, if any, should be based on the number on someone's driver's license. We know that if you've seen one 80-year-old, you've seen one 80-year-old. Every 80-year-old is different from every other. Some are very advanced in their aging. Some are very, very young in their aging. There are huge differences between people as we age, a great deal of heterogeneity, and this affects everything about that person's health. It's not too much further of a leap then, knowing that, as we all do, to realize that that's all grounded in biology. Aging is driven by biology, and it's biology that defines those differences between an 80-year-old who is playing tennis and playing pickleball and an 80-year-old who is fully functionally dependent in a nursing home. If aging is all driven by biology, we can understand it. We can understand the molecular mechanisms that drive these processes, and not only understand them and study them, but also manipulate them. This is aging biology. We often categorize these molecular mechanisms of aging by a scheme called the hallmarks of aging, meant to be as an analogy like the hallmarks of cancer. And some of these are more familiar than others, or more in the popular realm, like telomeres, or changes in protein folding that contribute to Alzheimer's disease, or changes in stem cell function. But these are all defined molecular mechanisms that we can study in the laboratory, and in every case, actually manipulate in the laboratory, genetically, or using drugs, and increasingly studying their manipulation in people in clinical trials using drugs. I think of, so how do you kind of make sense of that menu of molecular mechanisms? What drives aging? I think of these as grouping into three broad categories, which helps me understand what parts of aging are most amenable to intervention to improve people's health and improve their lives. So on the one hand, part of aging is driven by damage. Damage to our cells, damage to our DNA, damage to our proteins. This damage is an inevitable part of life. It is inseparable from the process of living. Our mitochondria generate ATP, and the process of generating ATP generates some oxygen-free radicals. You can't stop that. And those oxygen-free radicals will glom onto proteins and glom onto DNA and cause mutations. Damage is a part of life. It turns out, though, that damage doesn't really define aging. If you look at, for example, organisms that live a really long time, like we do, versus organisms that don't live very long at all, like the mouse, the little laboratory mouse with a 2-year lifespan, the rate at which they accumulate damage isn't really that different. And you really can't prevent aging or change aging by trying to slow the rate of damage. So it's there in the background, but it's not really driving what causes clinical effects. What is very different, if you look between us and a mouse, for example, is our capacity to respond to that damage and to repair it. So stress response pathways, repair pathways, protein refolding pathways, these are really the key to what makes for differences in longevity and differences in the rates of aging between species and probably between people too. These stress response pathways are also the most amenable to interventions because they're controlled by sensors in our cells. And if you can turn on those sensors or turn off those sensors, you can activate these stress response pathways. And most of the drugs that we'll talk about later that are being investigated as potential gerotherapeutics target these stress response pathways and turn them on. And the third category are consequences of all the rest. Kind of if you have enough trouble in one space, it creates even bigger problems unto itself. Mitochondria, for example. Mitochondria are the energy power plants of our cells. They turn sugar into ATP to drive everything a cell needs to do. Mitochondria get damaged. Their DNA gets damaged, their proteins gets damaged, they get less efficient at making ATP for our cells to use. And if enough mitochondria get damaged enough, then the cells don't have the energy they need to repair their DNA, for example. So mitochondrial dysfunction is an example of a kind of bigger consequence, a meta effect of molecular damage. Loss of stem cells is another example. If you have enough damage to enough stem cells, suddenly you can't repair a wound. You can't recover from a heart attack. So these consequences are another important group for designing interventions because if you can fix these big picture consequences, you can have an outsized impact on people's health and the effects of aging. So therapies that target stem cells, therapies to kill senescent cells are in clinical trials, for example. All of this builds up to the geroscience hypothesis, the big idea in the field, how all of this interesting biology might actually change the lives of lots and lots of people. And that is, given where I started with aging has a huge impact on how people respond to problems like broken bones. Aging has a huge impact on the risk for all sorts of chronic diseases. So if you can change that underlying common risk factor, interventions that target fundamental mechanisms of aging, you could prevent, mitigate, or treat not one problem at a time, but a lot of the different problems of aging all at the same time, rather than playing whack-a-mole with a whole bunch of chronic diseases. And we know that doesn't work very well. Multimorbidity is not just the sum of all of the chronic diseases that comprise it. Instead of playing whack-a-mole with a whole bunch of different things, you can change the underlying biology so everything might get a little bit better. That's the hope. We don't know if that's true. And for most of the last 15 years, this was kind of the out there, one day we'll figure out if this is real or not. But we're actually very close to testing this in clinical trials right now. And I'll tell you what those are in a few minutes. So that's a quick introduction to what is aging biology and what is geroscience. Geroscience, should have just said the definition. Geroscience is applying that aging biology to improve the health of people, applying aging biology to humans. Next, I'll talk a bit about my kind of personal career journey, but also what I've been up to the last 5 years in going from a basic science lab with a lot of mice and making mice healthier, living longer, that's nice, to wanting to change the lives of people and building tools and capabilities to do that. My basic science, many of you guys probably remember, is grounded in ketone body biology. I'll just give you a really quick introduction to that because it's just the jumping off point. But if you're interested in ketone body biology, feel free to look me up and look up some papers, and we're doing lots of cool stuff with mice. Ketone bodies are small molecules that your bodies all make, made in the liver from fats that are mobilized from adipose tissue. They circulate throughout the body. They're very simple molecules. This is the major one, beta-hydroxybutyrate, or BHB. After they're made in the liver, they circulate in the blood and are taken up by tissues that need energy. Most tissues, the brain, the muscle, the heart, kidneys, and most other things. And then they're burned to generate ATP. They are how you survive a long fast. If you're fasting for more than a day or so, most of the energy that your body needs comes from your adipose tissue, and a lot of that is in the form of ketone bodies, instead of glucose from carbohydrates. But you're always making ketone bodies, which is fascinating to me. You're always making a little bit, 24 hours a day. It's just you make a lot more, 100-fold more, if you're fasting or exercising. Ketone bodies are interesting from an aging perspective because fasting, exercising, caloric restriction, these are all things that activate those stress response pathways and have been tied very, very deeply to fundamental mechanisms of aging. And so we thought that ketone bodies are one of the mechanisms by which fasting, calorie restriction affect mechanisms of aging. And to make a 15 years of work into a very half a slide story, they do. Ketone bodies help to compensate for age-related declines in glucose metabolism. They're in clinical trials in heart failure and Alzheimer's disease for that purpose. But the focus of my lab has been understanding how they don't just get burned for energy, but they act like drugs, binding to proteins, altering enzymes, binding to receptors, and in doing so, acting directly on a number of these hallmarks of aging. If you're curious, you can look up some of the mouse papers that we did. We recently did a study showing that ketone bodies can facilitate the clearance of misfolded proteins from the mouse brain, that they serve a function of helping to maintain protein quality control in mice, maybe in people, but definitely in mice. And also that exposure to ketone bodies in the brain remodels the proteome of brain synapses, improves synaptic function, activates a particular signaling pathway called PKA. And these are mechanisms by which exposure to ketone bodies helps the mouse brain stay healthier in aging and function better in aging with better memory. So we're very good about making mice healthier. And through all this work with mice, we've learned that ketone bodies interact with most of these hallmarks of aging, which is really interesting. But probably not to all of you, and that's okay. And you know, I love basic science and mice are fun, but I'm a geriatrician and I want to help improve the lives of the people I treat. So from when I started as faculty, you know, my eye has been on how can I take interesting, important basic science and figure out if it's relevant to people, especially to older adults, and how can we apply it to improve the health of older adults. More than 10 years ago now, I got involved in a group of really smart people, all way smarter than me, basic scientists, clinical researchers, some geriatricians, many other specialties, an international group, all of whom were interested in the same question. How can we take that aging biology that we're all studying in the laboratory with mice and worms and flies and so forth, and test if it's actually relevant to people? How do you do clinical trials in aging biology? I was privileged to help to write some of these papers to describe, like, this is not a crazy thing. We think we can figure out how to run clinical trials to target aging. Over a few years, that eventually led to the formation of 2 national networks to facilitate the translation of aging biology into clinical trials. One of them is called the Translational Geroscience Network, funded by the NIH. Its role is sort of similar to like a cancer clinical trial network, meant to facilitate coordination, standardization of protocols, standardization of biomarker assays, real nuts and bolts stuff to make it easier to run early stage clinical trials that test ideas, test mechanisms, and provide proof of concept for aging biology interventions in people. The other one, which I'm one of the PIs on, is the Geroscience Education and Training Network. One of the main barriers to doing more clinical trials, more translational research in geroscience, is that there's not very many investigators who are familiar enough with basic science and familiar enough with clinical research and familiar enough with older adults to actually do these clinical trials do them well and know how to do them. So the Geroscience Education and Training Network is creating curricula for medical students and PhD students to help get people interested in the idea that geriatrics is an exciting translational field. And also for people who are a bit later at the stage of fellows or postdocs or junior faculty, to provide the kind of practical training that you need to be able to carry out a geroscience clinical trial. I'm happy to talk more about that later, especially if any of you might be interested in doing that training program. A lot of this is focused on running early stage clinical trials. This is the real bottleneck in the field. How do you get from mice to having enough evidence to run a practice changing clinical trial? First, you need to do smaller early stage proof of concept kind of mechanism focused studies to validate that the biology works the same way in people as it does in mice. It often doesn't. To test safety and tolerability, no matter, even if it's a drug that's in wide use in other people, you still need to test safety and tolerability and figure out what kind of dose to use in the older adults that you want to target. And hopefully generating signals of efficacy to help build evidence to justify doing a larger efficacy-focused clinical trial. For new drugs, this is the equivalent of phase 1, maybe phase 2 trials. But even if you're talking about a repurposed drug, a lot of you have probably heard about interest around metformin, for example. You still need to know, does it work the same in people as it does in mice? So even repurposed drugs, even dietary supplements need this sorts of early stage proof of concept studies. If I wanted to know if this ketone biology that I studied in mice was relevant to people, I need to figure out how to do these sorts of early stage clinical trials. Because what I really wanted to do was test whether ketone biology could help frailty. And I thought this made a lot of sense from a mechanism point of view. Obviously, frailty is a really important problem. I'm not gonna define frailty for you guys. Glad I don't have to. It's a problem without pharmacological therapy, and it's a hugely impactful problem. We don't know a ton about the biology of frailty. It's a great example, like many geriatric syndromes, of being understudied because it's thought to be too complicated, too many systems, too many organs, too many pathways. But if you distill this down to core molecular mechanisms of aging, I think it makes a lot more sense and feels a lot more tractable. So we think, for example, that some of the key features of frailty biology are reduced ability to metabolize glucose to generate energy. That's a common theme of many problems of aging, many diseases of aging. Dysfunctional chronic inflammation, and loss of muscle due to catabolism of muscle tissue. All of these three elements in mice are targeted by ketone bodies. They supplement energy, they help to dampen chronic inflammation. My lab had a nice paper about that, how they dampen chronic inflammation in the mouse brain, and they help to preserve muscle integrity in mice. So how can we start to test if this biology is actually relevant to people? First, we need an intervention. How do we give ketone bodies to people in a way that's feasible for a clinical trial? So we needed a compound. This is where we collaborated with this company that I co-founded called Component Health to develop a ketone ester. So this is a molecule that's essentially a delivery mechanism for ketone bodies. So you don't have to fast, you don't have to go on a ketogenic diet, you don't have to change what you eat. You're essentially eating a prodrug, more or less, that's rapidly metabolized into ketone bodies when you eat it. It's actually classified as a food ingredient because ketone bodies are normal biology, and it's broken down so fast that this molecule is undetectable in your blood after you drink it. So it's basically a delivery mechanism to eat ketone bodies. It's metabolized to ketone bodies mostly in the liver by two different pathways that I'm happy to talk about in detail for anyone who's interested. But the net result is you drink it and it generates ketone bodies in your blood. So this is potentially a feasible intervention for people to do, to take a dose of this once or twice a day. But before you launch into a clinical trial in frailty, what's the state of the literature for this sort of compound? And the answer a couple years ago was not much. Most of the research on ketone esters in general was done in young people. They're actually of interest for elite athletes for improving cycling performance, for example. That's where my colleague, collaborator, and partner in clinical research, Brianna Stubbs, got her start as a clinical researcher studying performance at Oxford. Now my colleague and research assistant professor at the Buck Institute. The longest study ever reported for one of these compounds was only 28 days. Never in older adults, and never testing anything about aging biology or frailty. So a lot of gaps to fill. And this is perfect for an early stage proof of concept clinical trial to start to fill these gaps so that we could move on to bigger efficacy focused clinical trials. This was the first clinical trial I did together with Dr. Stubbs, first clinical trial ever done at the Buck Institute. We figured out how to do this more or less starting from scratch, which was a really fascinating experience that I'm happy to share. Was actually a lot easier than I thought it would be, and had the benefit of letting us create, I'll talk more about this in a minute, but create a clinical research program that was perfect for what we want to do and perfect for the goals we want to accomplish. But our goals started very small. Fill some of these literature gaps around ketone bodies. So we did a 12-week randomized controlled trial with older adults in stable health, half of them taking a ketone ester, half of them taking placebo. The goals were pretty basic. We wanted to confirm kinetics. If you drink this much of a ketone ester, how high do your ketone bodies go? Most of all, we wanted to test safety and tolerability, specifically in older adults. Now, this is a food ingredient. It's sold to the public. It should be really safe, but there's no published data with older adults. So we wanted to fill that gap. We wanted to look for, see if we could detect signals of those molecular mechanisms that we've observed in mice. Can we see them in people too? And then hopefully look for a signal in clinical outcomes. So since these are not frail people, but it's meant to be preliminary data for a study in frailty. So we collected a variety of data on physical function, like short physical performance battery, gait speed, grip strength, 6-minute walk test, cognitive testing, and so forth. The long and the short of the results, most of this is published now. The kinetics are actually different in older people than younger people. They always are, but not enough to change how we would dose the ketone ester. We did fortunately safe and tolerable compared to the placebo. We didn't detect any change in physical or cognitive performance. Tiny study, healthy people. We weren't really expecting that. The signals of molecular mechanisms was interestingly reassuring though. So we did all sorts of fancy biology on these 30 people in the clinical trial, looking at their proteomics in their blood, their metabolomics, their immune cell subsets, cytokines, microbiome in collaboration with Peter Turnbaugh here at UCSF, looking for, can we detect changes in proteins or mechanisms that are similar to what has been reported in mice? And we actually did. None of this is published yet because it's a huge amount of data that we're still analyzing. But for example, among the proteins that change the most in people taking the ketone ester versus placebo, some of the very top hits are proteins that have been reported in the literature to be targets of ketone bodies for changing aspects of biology. And similarly, immunophenotyping, looking at T cell subsets, some of the T cell subsets that change the most in people taking the ketone ester versus placebo are T cell subsets that have been reported in multiple papers to be regulated by ketone bodies in ketogenic diet. So we actually are seeing signals of similar biology, which is very reassuring. And help to support and justify moving on to what we really want to do, which is a larger, multi-center clinical trial enrolling people who are pre-frail or at an early stage of frailty and testing if the ketone ester can improve their function. So this is called TAKEOFF. I came up with that acronym all by myself without any help from an AI, targeting aging with a ketone ester for function and frailty. And that's exactly what the clinical trial is testing. Using lessons from our preliminary pilot study, it's bigger, it's longer. We're using a higher dose now that we know it's safe. We've changed a few other things to optimize our ability to detect difference using a composite primary outcome of multiple quantitative measures related to frailty that will hopefully give us a bigger overall picture of the change in people's function. And this is funded by the NIA and a multi-center study coordinated by the San Francisco Coordinating Center, Peggy Cawthon and Steve Cummings, and involving collaborators at UConn, George Kuchel, Jenna Bartley, and Jeff Folick at Ohio State. Takeoff really launched. We really kind of actually, it was a bit of a bumpy takeoff for Takeoff, but we officially got off the ground in the spring, and Takeoff is well going. Enrollment is actually going pretty well, and we're hoping to be finished in about a year and a half. And that's all happening at the Buck Institute. Buck Institute has never done clinical research before. Everyone, of course, in the aging biology world knows it as a world-class basic science institute. Lots of mice, lots of flies, lots of yeast, lots of worms, lots of fancy machines. It had never hosted any sort of clinical trials. But we built a clinical research center gradually, starting from that first pilot study and expanding very fast over just the last 2.5 years. Our clinical research center is now housed in a suite and actually half of a floor in the main Buck Institute building. We have, for example, a beautiful participant lounge for people to hang out in. We can do sophisticated functional testing, including VO2 max cardiopulmonary exercise testing with older adults, all sorts of strength testing, cognitive testing, blood draws, muscle biopsies, all sorts of stuff, which enables, and because we're located at a place with world-class basic science labs, literally down the hall and up the stairs, we can combine clinical research with older adults with really fancy biology. And I'll give you some examples of that. 3 years ago, this was me and Dr. Stubbs and a couple of RAs in my lab who mostly did Western blots, but were curious to get into clinical research. And now this is our team. There's probably someone I've already forgotten to add to this. Our clinical research group is now much bigger than my basic science lab, having grown very fast. And this is a joint group with Dr. Stubbs, to be clear. We're a very diverse group. We're a multilingual group. We conduct study visits. Takeoff is being run in both Spanish and English, for example. We have research nurses. We have a number of part-time research physicians besides myself. Postdocs, recruitment specialists. It's an amazing team, which is growing fast. Because one key lesson I learned is that clinical research infrastructure is not about places, and it's not about stuff. It's about people. It's about finding good people and building a good culture and learning how to maintain people. Why are we doing clinical research at a basic science institute like Buck? And I get asked this question a lot. Why aren't we doing clinical trials at a hospital? Because the goal is to do those first steps into human biology that can't really be done anywhere else, and that will lay the groundwork for bigger studies, practice-changing studies. So basic experimental studies in humans, this is kind of the core focus. This is doing basic science with people rather than with mice. We need to do a lot more of that. And I'll give you a couple of examples of studies like this that we're doing right now, besides that pilot and Takeoff. Early stage trials of interventions. Takeoff is a great example of this. We're doing a couple of others where we're basically seeing do compounds, do interventions work the same way in people as they do in mice. And we want to be a node for larger multi-center clinical trials. Takeoff is a multi-center clinical trial being led by the Buck, but one of 3 enrollment sites. And I'll talk to you about 2 other examples of national or international groups where we're hoping to be a site in a larger effort. The first study, I'll give you 2 kind of thumbnail sketches of what kind of science are we doing. One is called MOVE, Molecular Optimization via Exercise. There's a lot of literature about how good mobility and activity and exercise are for all sorts of aspects of health. The primary part of interventions to try to prevent Alzheimer's disease is always about exercise. Try and prevent frailty, always about exercise. Exercise helps pretty much everything. Why exactly? That's all biology too. It's biology that's hard to understand because it's complicated. But if we can figure out what that biology is, then we can provide those benefits to everyone, whether or not they exercise, whether or not they're able to do activity like that. What are the most important molecular benefits that we get from exercise? But to me, the opposite of that is also really interesting. You can exercise a ton, you can do all the right things for all of your life, but still have serious health problems as you get older. Nothing is great for everyone. Nothing works the same way for everyone. So what does exercise not do? What parts of aging does it not affect? And so, even if we're able to provide the effects of exercise to some people through molecular mechanisms, what are we missing? We want to answer both those questions. What might be the most dramatic way to get those answers? Well, maybe study people who have been exercising all their lives in the most intense possible way. Elite older adult athletes. So MOVE is an observational study of people over 65 who are elite level competitors mostly in endurance sports, so running, cycling, things like that, compared to a control group of people who are basically in good health, but are basically sedentary. So what are the most dramatic changes we can observe between these elite older adult athletes and normal people? And besides doing exercise testing with them and measuring their grip strength and all that sort of stuff, we do muscle biopsies and take blood samples. Because we're right next to a basic science lab, we can do all sorts of fun things with these muscle biopsies, like looking at 3D histology, looking at their mitochondria under electron microscopy, measuring the function of their mitochondria using fancy respirometers, stuff that you can really only do if you're walking the sample right to the lab. And the most exciting thing to me, you can grow muscle cells in culture. And then you can turn those muscle cells into little muscles in a test tube, basically. You can grow little muscles in test tubes. Not just grow them, but you can hook them up to little tiny electrodes, zap them, so our muscles activate when our nerves deliver electricity, right? So you can do that in a test tube. You can exercise these muscle organoids in a test tube. Exercise them as if they're running a marathon. And because they're in a test tube, for example, you can do mass spectrometry proteomics on everything that they're releasing as they're exercising. So you can get really detailed biology on exactly what happens when a muscle is exercised, what might be the molecular mediators of the effects on brain health, for example, and see how these are different between muscle that comes from a highly trained older adult athlete and someone who is sedentary. So MOVE is an example of basic science with people that hopefully will lead in the long term to interventions that can help improve the lives of millions of people. Time is another example. Temporal investigation of multimodal elements. Who's ever ordered RNA sequencing or proteomics in their clinic? Nobody in this room and nobody on Zoom. These are research tools. In the lab, we can measure everything. We can measure hundreds of thousands of things from a blood sample, tens of thousands of proteins, tens of thousands of metabolites, small molecules. We can measure all the RNA strands, all the DNA, all the cytokines, all the immune cells, hundreds of thousands of things. The biggest reason why this is still a research tool and none of us order them as physicians is we don't know how to interpret all those data, especially if you just have one blood draw. You have 100,000 things. Some of them are high, some of them are low. How do you know, is that part of a trajectory? Is it going up? Is it going down? What does high even mean? What does low mean? Is it high because of what they ate for breakfast? Is it high because they're about to get cancer? We have no idea. So these sophisticated tools are not terribly useful in clinical practice. Time is meant to change that. Time is enrolling about 150 people who will be the most heavily characterized human beings ever. We're measuring hundreds of thousands of things, all this stuff, at 44 time points over 11 weeks, every couple of hours over the course of several days, every couple of days, every week, after they exercise, after they do a glucose tolerance test, and not just the blood stuff, but all their microbiomes from every piece of their body, all of their activity, their grip strength, their cognitive tests, all sorts of stuff. And working with a big AI-driven machine learning data science team at the Bucks Institute, trying to put all of this into a big machine learning blender so that we can actually understand those 100,000 things. What do they all mean? And if we can understand what they all mean, then we can actually derive useful information, clinically relevant, clinically actionable information from that. So TIME is funded by the Advanced Research Project Agency for Healthcare, ARPA-H, which funds blue sky, big deal, world-changing things. And the big idea is that hopefully TIME will help us take these sophisticated research tools and actually put them into the hands of clinicians to improve care of our patients. I think, so I'm very proud of what we set up. I think this is an example of what our field needs more of. Clinical research centers that specialize at this interface of geriatrics and basic science, closely linked to basic science laboratories, expert in the care of older adults specifically, in how to measure, how to interact with older adults, how to measure things functionally, and deeply engaged with our local community, partnering with senior centers, partnering with geriatric practices, talking to older adults about what they want out of research and what questions we should be asking. And the big picture, bringing geroscience into clinical practice. So the last bit here is, what is the overall state of the field? Clinical trials that will change the way that we, as geriatricians, care for older adults, still seems like science fiction, but it is rapidly becoming real. There are hundreds of these early stage clinical trials happening all over the country, all over the world now. The first time I gave a talk about aging biology to fellows, 2013-ish, there was one. That was the very first one. That was this one right here. And now there's hundreds. And I'm doing some of them. I already mentioned these two national networks to support clinical trials and to support training of investigators to carry out clinical trials. I mentioned drugs a couple of times. Do drugs actually exist that target aging and that could fulfill that geroscience hypothesis? And yeah, so there's a lot of people doing drug screening on all those fancy mechanisms, trying to come up with new compounds, new drugs. But what about drugs that are already in use? Are there any that might actually target aging mechanisms? And could we find out whether they do? So these are drugs that in preclinical models like mice increase the health span, the lifespan of healthy mice. Maybe they work across multiple disease models. And in clinical trials, what might you expect to see if there were a drug that is actually a gerotherapeutic? We just don't know it yet. Well, it starts off, it gets approved for treating whatever disease its drug company proposed it for. So it improves blood sugar control, type 2 diabetes. All right, so now it's on the market as a diabetes drug. But then in the next clinical trial or in observational data, it's shown to reduce mortality in diabetes. For a long time, there was only one drug clearly shown to reduce mortality in type 2 diabetes, metformin. That's now changed. Several of the newer agents also reduce mortality, but that's not obvious that all drugs that treat diabetes would actually lower mortality. Maybe it's also shown to prevent cardiovascular disease. One of the large clinical trials of a newer agent in tracking adverse outcomes tracked cardiovascular events and turned out that actually cardiovascular events were not more common in the drug group, they were actually less common in the drug group. And that led to a large clinical trial specifically of that drug to prevent cardiovascular disease in people with diabetes. Oh, sorry. Yeah. I'll just unplug and replug. That always works. Okay. So you find it reduces mortality. Then you find that it doesn't just treat diabetes, it treats comorbid diseases that commonly happen in people with diabetes. Like it helps to treat heart failure, helps to slow the progression of kidney disease in people with diabetes. Then you find out that it helps to slow the progression of kidney disease in people who don't have diabetes. You don't actually need diabetes at all. And then you find out that it prevents or treats multiple diseases in people who don't have diabetes, aren't obese. If you get all the way to the bottom there, so it turns out that drugs work for multiple chronic diseases beyond the indication it was really approved for, you probably have a gerotherapeutic. No drug has gotten all the way to the bottom yet, but several have completed several of these steps. Some of my colleagues at Albert Einstein, Sandra Alexic and Nir Barzilai, have tried to quantify this for what are the most promising potential gerotherapeutics based on molecular mechanisms, mouse data, and human clinical trial data. And some of the ones that rise to the top are SGL2 inhibitors, metformin, the GLP-1 receptor agonists like Ozempic. And interestingly, one of the bisphosphonates too, specifically zoledronic acid, not other bisphosphonates, just that one. So these are at the top of the list, mostly based on clinical trial data of potential gerotherapeutics. But how would we know? So what clinical trial would you actually run to see if, well, will this actually target aging and prove the geroscience hypothesis? What will your outcome be? There's a few good examples of, or a few good ideas for this. That question, what would be the outcome of the clinical trial, is often, if you just ask that of a random scientist or a random person, sounds pretty crazy. So how do you run a clinical trial that targets aging? Do you run a clinical trial for 100 years and see if people live longer? Good luck getting that funded. But fortunately, there are other options. Some of the best options, I think, include geriatric syndromes, like can you improve frailty? But also multimorbidity, using a MACE-like composite of multiple chronic diseases as your primary outcome. This was the primary outcome of a large clinical trial plan to test whether metformin could delay the progression of multimorbidity. That's still a really good idea and should be amenable to the FDA as an indication for a drug. Lately, though, I think the top candidate for what will this landmark clinical trial wind up actually capacity is, and why that might actually be an indication for us to start prescribing drugs within 5 years. Intrinsic capacity is it's more of a global concept than a US concept, championed by the World Health Organization. It is the composite of all of the physical and mental capacities of an individual. It's basically a way of quantifying a comprehensive geriatric assessment. So its domains include cognition, mobility, sensory function, psychological health, and vitality, nutrition, strength, things like that. You can measure and quantify all these things in different ways, as I'll talk about in just a minute, to get a comprehensive picture of the overall state of aging, state of health of an older person. Intrinsic capacity is then paired with interventions that target those different domains with the idea of improving the health of an older person. That's called ICOPE, Integrated Care for Older Persons. And this is a global framework for improving the function of older adults based on systematically targeting the domains of intrinsic capacity. Keep those in mind. You can measure intrinsic capacity across the lifespan. So it's not just for older people. It's easier to measure in older people because of that heterogeneity in aging. It's easier to detect the decline. But you could, if you measure in sensitive ways, you could detect changes in intrinsic capacity earlier in life, even in middle age, or even before. It depends on how you measure it. If you measure it in sensitive enough ways, you could, for example, detect whether your intrinsic capacity is starting to decline, not just when you're 80, but when you're 60, when you're in otherwise good health. So you can operationalize intrinsic capacity in different ways for different contexts based on the measurements that you're doing. The most common way to measure intrinsic capacity so far is using classical geriatric measures, like 3-atom recall, chair rise, things like that. That's great for older adults. But part of the promise of intrinsic capacity as a concept is if you come up with more sensitive ways, you could detect changes earlier in life and provide interventions that are helping to prevent problems of aging from occurring at all. This is relevant to drug trials because intrinsic capacity has a diagnostic code in ICD-11. And insurers and Medicare and the FDA love diagnoses. So there is a diagnostic code, MG2A, for aging-associated decline in intrinsic capacity. And the big idea is that that could become an FDA indication for a drug, if you can figure out how to measure intrinsic capacity in a careful enough way. So first, intrinsic capacity. I want to talk briefly about the X Prize health span, because we're taking an intrinsic capacity focused approach to trying to improve the health of older adults. X Prize is a foundation that provides big cash prizes to incentivize groups to do audacious things. The audacious thing that the X Prize health span wants to do is restore 20 years of function in brain function, immune function, and muscle function with a 1-year clinical trial. It's a global competition that's ongoing right now. It's being led by Jamie Justice, formerly faculty at Wake Forest, who is a very classically trained epidemiologist and aging scientist. It's a very rigorous and well-designed study or competition, and it's elicited interest from groups all over the world. We have an X Prize team with the University of Toulouse, which is unique and I love because it combines the best of geriatrics with geroscience and aging biology. So Toulouse pioneered what they call ICOPE Intense. Remember ICOPE, Integrated Care for Older People, intrinsic capacity focused multi-domain intervention, exercise, nutritional counseling, meditation and mindfulness, cognitive training, focused on frail older adults. And they've already shown that ICOPE can improve the health of older adults, and they want to see if they do this training as intensely as possible, how much impact can that have? We're adding to that drugs and possibly supplements that target the biology of aging to try to have the biggest impact. Toulouse is running a pilot clinical trial right now. We're partnering with a senior living group called Human Good to implement a version of this in the Bay Area, run out of the Buck Institute, involving residents of affordable housing and independent living communities in the Bay Area to do an ICOPE Intense style intervention along with drugs and some clinical interventions. This trial hopefully will start next summer. It's in an advanced stage of design, partially funded. We're hoping to secure the rest of the funding in the next 6 months. And I'm mentioning it to you because we're looking for partners. We're looking for junior faculty, clinician scientists, fellows who might be interested in helping to lead and run a clinical trial like this that combines the best of geriatric care with geroscience. The last thing is another ARPA-H program called PROSPER. The goal of PROSPER, ARPA-H similarly funds audacious concepts. The audacious concept is to get to a drug approval for age-associated decline in intrinsic capacity within the next 5 years. Their pitch is that, and I love this. Right now, if you're in your mid-60s and not feeling as great as you used to, you go to your doctor. Your doctor is going to tell you there's nothing to do. That's not true. I know that's not true. This wasn't written by geriatricians. This is written by people who don't really understand what the care of older adults is like. But it's a very kind of morbid picture of the current state of healthcare. Wait until you get really sick and then you get prescribed a bunch of drugs. But the vision for the future is you'll be able to detect that you're beginning a decline, and then there'll be a variety of interventions with FDA approval to slow and reverse that decline. And so you'll enjoy an extra 20 years in good health. And that is a nice vision. Geriatricians need to be involved in this stuff. A lot of these sorts of efforts say things like, we want to make geriatricians obsolete. I don't think they'll make geriatricians obsolete. I think this is what we need to do. Yeah. So I'm part of a team, and we're hoping to get to do this. This is my last slide. To accomplish some of the goals that will lead from starting from scratch to a drug approval in 5 years. So first, you need to figure out how to measure intrinsic capacity in a cheap, easy to use way that any person in the US can do at home in 5 minutes and cost less than $100, because you would do this just like you would check blood pressure. Validate that kit in a 1,000-person, 3-year clinical study across a range of ages, including an intrinsic capacity-based multi-domain intervention to improve people's health, to show that your measurement gets better as people's health gets better. This is just the starting point. These are hugely ambitious studies already, and it's just getting warmed up. And that's just to generate the kits as a outcome measure that can then be used in a phase 3 trial of a repurposed drug like metformin or a GLP-1 receptor agonist to generate an FDA indication. If this sounds interesting to you, hopefully my team at the Buck Institute may get to do a piece of this in the near future. And if it sounds interesting to you, feel free to reach out because we'll be looking for help. So the future is now. If you want to learn more about how to get training to do Geroscience clinical trials, let me know. If you're interested in learning more about our clinical research center, collaborating, running part of your studies there, using us as a site, let me know. And there's a lot of exciting stuff going on around the world. The first drug with an FDA indication for age-associated decline in intrinsic capacity will probably be approved in the next 5 years. I think more likely than not. And all of this takes a lot of funding and a lot of great collaborators and a lot of work, especially Dr. Stubbs, the co-leader of our clinical research efforts at the Buck. So thank you all on Zoom, and thank you guys for sticking around.
Does anyone have any questions for Dr. Newman? We have one on Zoom from Alex Smith. Machine learning is old AI. Some think new AI will lead to breakthroughs, others skeptical. Do you believe new AI will transform geroscience? Also, where did you take that background beach photo?
What's the name of your dogs?
Olivia and Fiona's favorite place in the world is Fort Funston, right in San Francisco, the greatest dog park in the world. Yeah, so I'm not an expert in AI. I have hope that sophisticated algorithms and new ways of doing AI will help to figure out how to make sense of that incredibly complex sets of data to derive meaning from giant data sets like time. Ultimately, everything has to be validated afterwards. And I think that's the key thing. Whatever interesting, fascinating results come out of these large analyses have to then be tested. And hopefully I'll get to do some of that.
Thank you, John. That was so awesome. Thank you for that fantastic talk. And I'm not going to ask you a question. I can just comment. I mean, one thing you convinced me of today. So I started out thinking at one point I was going to be a basic scientist, and then I wasn't very good at it, and I've run away in my life ever since. And I always viewed epidemiology and outcomes research as kind of my escape. And you've actually done more than anyone. It's sort of making me realize, I can't really escape anymore. I mean, because these things are going to become important across the realm of research and medicine. And I mean, and one of the things I just found fascinating about your talk is you introduced a lot of basic science concepts that are new to me, or molecules that are new to me, and you spoke about them in the same way that I could talk about any risk factor or predictor of outcomes, or so it's fascinating how this is all coming together. So thank you. So
we seem to have a special skill in our division at creating the translational epidemiologists. A couple of examples of which are pairing basic science concepts with epidemiology and aging.
Thank you, John.
Thanks, guys.